So my role is really focused on microbiology. As you can see from this new beautiful lab, we now do all sorts of experiments related to monitoring bacteria and pathogens, viruses as well. We are definitely setting up more experiments in the area of water and wastewater treatment. And we're trying to sort of expand into other areas to support the PFAS research in terms of bioremediation and so on. I like to pride myself on trying to communicate science in a way that helps our clients make the best decisions possible. The CDM Smith Research and Testing Lab is the laboratory facility where we house both our chemical and microbiological testing. It has equipment, dedicated staff, and the capability to really do a very broad range of analytical testing for clients. We have the ability to have samples come in and just analyze those in-house, but we also have enough space to potentially do small pilots if we need to. The results get synthesized by our Bellevue Research and Testing Laboratory staff, and then basically presented to the client so they can make better decisions. When you have a lab in-house, it really makes your research independent of the wills and interests of your collaborators and the people performing the research. It allows us here at CDM Smith to really step back, ask the real questions our clients are interested in, and create the data that is most valuable to them to make their decisions. So to give you an idea, I used to be a utility scientist for 10 years, and one of the things that scared utilities the most is technologies that are very, very novel. The thing people say is you want to be as innovative as possible, but you do not want to be on the bleeding edge of technology. So being independent, being here to inform our clients really allows us to transition that science, those innovations and technologies, understand when they work, when they're going to be challenged, and basically make it work in the field for our clients in the best way possible. When you look at the water cycle as a whole, microbiology has a role to play in every single part of that. So starting at the drinking water, source water side, you've heard of harmful algal blooms and some of the changes we're seeing in terms of cyanotoxin contamination of water bodies. So one of the things we can do in this lab is actually looking for those toxins and looking for the organisms that produce them, right? So that's a pretty critical function where we can support our clients, especially in the design phases of some of their plants and some of the upgrades that they're looking for. On the wastewater side, we've really expanded quite a bit. We've been really trying to get more utility clients to go back to activated sludge analysis. This is where you look at your wastewater sample from your bioreactors and you figure out if there's a bad filament in there that causes that foaming and bulking a lot more in your process. We're trying to see how we can optimize those processes to reduce the biological upsets that are happening. And then there's the most interesting and probably the most exciting area, and that's the reuse and desal area. And as you can imagine, when we talk about potable reuse, the most important part is making sure that water is safe. So we have lots of really cool projects, starting with the Los Angeles Department of Water and Power, Metropolitan Water District of Southern California. We're looking at the product of these advanced treatment plants to make sure they're safe and virus-free. So I had a love for science, and that love of science pretty much drove my instinct to try and solve problems and find solutions to things, right? But I think more importantly, when you're looking for solutions, a lot of the time it's easy to fall into the same ideas, into the same pace. And microbiology is not that at all. Microbes are the original survivors. They manage to do things that we are just not capable of. They adapt to environments, and when they switch on or off a gene or use a resource, we can use that to enhance technologies, to change our environment, to modify it. And I think that level of complexity and just that potential made me love microbiology. That's right. There's a lot of new technologies that are very, very interesting. Here at CDM Smith, we've really invested quite a bit of time and effort into understanding hydrocyclones, right? So that process is called densification. It has a very small footprint, easy to upgrade to, requires a lot of operating know-how, but we've got that at CDM Smith. recently, there's been quite a bit of hype around the idea of nanobubbles, and those are very, 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 very tiny bubbles. So you may be going, well, we pump air into wastewater systems all the time. What makes these different? And the idea is that once you hit a diameter that small, then you are literally defying the laws of physics that apply to larger bubbles. So instead of these bubbles rising to the surface, they stay suspended. And they avoid the buoyancy issue, which means that you actually increase your oxygen transfer rates. So you're breaking down more organics. You're potentially impacting some of the pathogens in the matrix as well, and you're nitrifying. We had the capacity to actually go out and do a nanobubble trial at a facility in the southern United States. We invested our internal funding because we were just that excited about understanding how the process works and what the limitations are. So we really pushed it to try and understand that a little bit better. So the results are actually quite complex, but we're really excited to be able to share the results in the near future. But there is an awful lot of interesting technologies out there as well, where there is the potential to really reduce greenhouse gas emissions or energy consumption, like MABR, for example, right? Membrane aerated biological reactors. Those are technologies that I think we as an industry are really excited about and would love to see where they go from here.
That's right.
We tailor solutions traditionally by really understanding the problem that the client is facing. And it tends to be one of two things, usually when you're talking about microbiology. We tend to have good and bad bugs, regardless of how you look at the problem. But let's say you're looking at your final effluent at a wastewater treatment plant, or your treated drinking water, or whatever that is. Suddenly the good bugs are just anything that doesn't cause disease or that is an indicator organism. So in that case, let's say the client wants to know how to monitor drinking water better in the distribution system. We can go out, we can understand what their level of risk aversion is. If they're willing to adopt a new technology, we can start talking about some of the tools that are out there, like adenosine triphosphate testing, for example, which only takes 5 minutes to test for versus our traditional 24-hour tests. But if you're talking on the wastewater side of things, suddenly the good bugs are the ones that remove nutrients, that actually help us perform better. And in those cases, basically, we're trying to figure out whether those bugs are present and whether the conditions around them facilitate their growth and their metabolism. We can use the qPCR to tell them if they're there in the first place, but then we can also use some growth-based tests to tell them, well, you know what, they're there, but they're going to need some additional resources, whether that's carbon or DO or anything else that those bugs need. I actually oversee Water Research Foundation Project 5209, Give Membranes the Credit That They Deserve. And this study is specifically looking at low-pressure membranes, so that's microfiltration and ultrafiltration, and how we can help our clients actually get credit for them in reuse scenarios. So right now, if you're looking at most of the regulation around the country, membranes don't actually get credit. And the reason for that is we don't have a good way to do daily testing to confirm that the viruses are actually meeting their log removal targets. So what we did here at CDM Smith is we introduced a portable, field-friendly PCR, and with a variety of adjustments, we're able to reduce the time it takes from potentially 12 to 14 hours to run for 5 targets, all the way down to 5 hours. So in practice, that would mean if you have a plant somewhere in California, an operator could potentially collect that sample and run the qPCR by the end of the day without having to go home unsure what the quality of that water is that is reaching the customers. I think science is one of these interesting areas right now where there's been a real tension. Technology has been absolutely accelerated at a pace that we've not seen before, especially when you think of COVID in the last 4 years. But at the same time, there's been a little bit of mistrust in the public when it comes to science. And that tension creates a really interesting dynamics where our clients are sometimes caught in the middle trying to understand what it all means. So I often say that the most important part about running a lab and producing data is really making sure that science communication is tip-top. We want to make sure the science is just as good as the communication side of things. And that sometimes means actually trying to show the client what they're seeing. There is nothing as impactful as when a client comes to you and says, you know what, my plant stinks, it's bulking, it's foaming, the operators are complaining, what do I do about that, right? And we can absolutely put that slide on the microscope, we can run that sample by qPCR, and we can give them real instructions on what to do next.
That's right.
Collimated beam testing is one of the services we offer to our clients. It's actually figuring out the dosage of UV required for us to be able to eliminate indicator organisms like E. coli and total coliform. So when you're doing a design for your plant or you're optimizing your UV system to disinfect wastewater effluent, you can send us a sample and we can test it for you to let you know. The other thing we do sometimes to help the clients actually figure out how to design their plants or how to optimize their primary treatment process is we do jar testing quite a bit. So that's something we can offer both drinking water and wastewater plants, where we take our samples, we essentially try to mimic the process of flocculation and sedimentation, and then that will inform us what conditions result in the best sedimentation process, so that primary treatment becomes a lot easier for the clients, because it's not as easy when you're dealing with millions of liters of water in giant basins.
That's right.
I think a lot of people are still asking the question of, what are the regulations? Are we making sure that water is safe? And I think California is leading the way on that right now. When it comes to scarcity, I think we're at a point where nobody can deny the need in terms of water reuse. Where I think we as CDM Smith and as the water sector are making a huge difference is, this isn't new to us, right? We've been reusing non-potable water for a very long time for agriculture, for industry. Where I think the major challenge comes in now is that science communication piece, where we can explain to the public that we are actually doing all the testing we can to confirm their water is safe from organics. We're looking at viruses, but we're also looking at cryptosporidium removal, which are protozoan oocysts, right? They're very resistant and you cannot chlorinate them. So we actually do look at the impact of removal, ensuring that everything is removed in that train. And I think ultimately there's also a lot of questions about the distribution of reused water. So you want to make sure you don't have regrowth, you don't have recontamination of that water once it leaves the plant. And that's another area where we as sort of an industry are trying to tackle the monitoring tools we already have available to make them more timely and cheaper and more efficient and field friendly.